Thank you, Richard. So good afternoon, everybody. As Richard mentioned, my name is Sarah, and I'm a PhD student based out at the Hudson Institute. And today I'll be presenting some of the findings to come out of my PhD. And my talk is titled Identification of Therapeutically Sensitive Molecular Dependencies in Diffuse Intrinsic Pontine Glioma. So there's been a few talks already about DIPG, but for those of you who are a little bit more unfamiliar about it, I'll just give you a brief overview. So DIPG is a pediatric high-grade solid glioma that arises within the midline, primarily within the pons. These patients are typically quite young. They're about 6 to 8 years of age when they're initially diagnosed, and they have a series of very rapid onset of symptoms, such as cranial nerve palsies, motor neuron weakness, and cerebellum dysfunction. A diagnosis is usually made by radiological imaging, and again, it has a very set imaging characteristics that I've listed here. Unfortunately, patients who are diagnosed, they do start the standard care of therapy, which is really limited only to radiotherapy, and this really doesn't improve their overall survival, and they have most patients who succumb to their disease within one year from initial diagnosis, and there's a 5-year survival rate of approximately only 2%. However, within the last decade, we've seen a significant increase in our understanding of the molecular underpinnings of DIPG, in particular the identification of an oncohistone mutation known as H3K27M that can arise in either the H3.1 or H3.3 variant genes. This mutation is a missense mutation, and it causes the loss of the trimethylation markers by inhibiting the PRC2 complex, and this induces a hypermethylated state of the chromatin, but it's not the only mutation that drives tumorigenesis. We do need co-occurring mutations such as in the TP53 mutations and also other growth factor signaling mutations to really drive this disease forward. So our group has developed, so we know that DIPG DMGs have a very diverse molecular landscape, and they consist of lots of targetable molecular dependencies, but these all have potential therapeutic exploitation. And our group has developed a functional genomics pipeline to identify these targets for therapeutic exploitation. So the Victorian Pediatric Cancer Consortium has developed a childhood cancer cell line atlas, and this constitutes a variety of different pediatric cell lines, high-grade gliomas, ATRTs, medulloblastomas, osteosarcomas, and we have a small subset of adult high-grade gliomas as well. So this figure is just showing the DMG-DIPG cohort that we have available. The data set includes whole genome sequencing, RNA-seq, methylation array analysis, and we've recently started including our proteomic work as well. And I've just listed what data sets we have available for the appropriate cell lines, such as the functional genomics screening with our CRISPR-Cas9 loss of function screen and a high throughput drug screen as well. Now, this was a massive collaborative effort, both internationally and nationally, and I really do have to acknowledge everybody who's been a part of this project, because we really wouldn't have been able to get this far without them. And this atlas is currently under review, and it was led by 2 of our senior postdocs within the program, Dr. Claire Sun, who's here today, and Dr. Paul Daniel. So this is a very brief schematic of our functional genomics pipeline. So our pipeline consists of probably 3 major arms: omics profiling, therapeutic screening, and CRISPR-Cas9 genetic screens. So our patient-derived cell lines all undergo multi-omic analysis, where we look at their mutational burden, gene expression, and also methylation array analysis. We then take these through high-throughput drug screening, where we look for drug sensitivity, and we also employ in silico blood-brain barrier prediction models. And finally, we use a custom 300 gene target pooled library to discern druggable genetic dependencies. So we can combine all these data sets together, and this enables us to identify enriched oncogenic pathways and genetic dependencies and their associated molecular inhibitors, which we then carry forward into our secondary in vitro and in vivo validations and also combination investigations. So this is just an overview of the high throughput screening. So we have on top our CRISPR screen data. This is a loss of function screen of over 300 genes, and it's represented by 1,200 guide RNAs. And you can see the number of cell lines that we have managed to screen, and also the various genotype. And along the bottom here, I've got the high throughput drug screen. So we've got a few more cell lines that have managed to get through drugging rather than CRISPR screens. And then on the end of each slide, I've got just the number of hits that we were able to identify. So our CRISPR-Cas9 screens, we were able to reveal specific genotype-specific hits, but also hits that were shared amongst the 3 different cohorts of our 3.1, 3.3, and our wild-type groups. In our drugging cohort, we looked to see what molecular inhibitors were really sensitive in our DIPG cell lines, but also had little to no effect in a non-neural stem cell control. And those are the ones that we kind of took forward for our secondary validation. So we combined this data to see where the overlap was. So we wanted to find molecular targets that while had a therapeutic sensitivity, but also were a tumor genetic dependency. And by doing this analysis, we were able to reveal 22 such targets. But we wanted to really know whether or not are these pediatric specific targets or are they generalized to other cancer subtypes and also to adult studies as well. So we compared these targets that we identified to the adult CNS tumor cohort in DepMap, and we looked to see whether or not these hits were also dependencies in the adult analysis. And we actually identified in the top left-hand corner here a number of hits that were very specific to our DMG-DIPG cohort, but had little effect in the adult CNS group. And this really does highlight that these are very specific to our pediatric samples. And when we looked at the pathway analysis for these hits, we wanted to see, are they in any enriched DMG-specific pathways? And there are several of them that are well known, such as the PI3K and AKT signaling pathway, TP53 transcriptional regulation, to name a few. There's probably a few that are not as well investigated in DIPG or DMG just yet, such as simulation, of which is a post-translational modification. So I'm now just going to speak a little bit of our validation of some of these targets. So I have, for my part of the program, I have chosen 6 touch targets to investigate further and validate and hopefully look for combinations. But I'm just going to talk a little bit about this HDAC2 dependency that came through in our cohort. So we identified that our H3.3 mutant cell lines were really dependent, had a really strong cell survival dependency for HDAC2, but this wasn't observed in our H3.1 cell line. So you can see here we compared the Z-score threshold of our genetic dependency, which we have a threshold of less than negative 1.5. So our 3.3 lines sit well below this threshold in this density plot, whereas our 3.1 lines, you can see it's a little bit more spread out. When we also looked at the HDAC1's role, because HDAC1 and HDAC2 are quite similar and have similar homologue of each other, when we compared these two genes in terms of the different cohorts, we didn't see any significant phenotypic effect for knockout of HDAC1 across any of our DMG cell lines, and there was no significance between the H3.1 and the H3.3. Conversely, though, the H3.3 mutant cell lines had significant dependency on HDAC2, and this wasn't observed in our H3.1 cell lines. So to look to see what this phenotypic effect was, we did a CRISPR-Cas9 arrayed knockout screen where we looked to see whether was this a cell survival dependency or some other dependency associated within the cell line. So we found that our H3.3 cell lines had significant loss of survival after transduction of HDAC2, whereas we didn't observe this in our H3.1 cell lines. And to look to see whether or not this was due to HDAC2's catalytic effect, we actually did a rescue assay where after knockout of HDAC2, we transduced with either a wild type or a catalytic dead mutant HDAC form, and we found that the wild type was actually able to reverse the cell viability and kind of rescue that function in there. But we know that a single arm of therapy is insufficient to really be as a treatment strategy for DIPG, so we want to investigate other combination arms. So going back to our list of drugs that had significant genetic dependency from our CRISPR and combined CRISPR and drug screens, we looked to see whether any of them actually had blood-brain barrier predictions. So we've utilized 3 in silico prediction models, which I've listed and cited here. And out of these, we had some novel drugs that probably have been not as well investigated in pediatric brain tumors. One of them was prasinostat, which is a pan-HDAC inhibitor, and onelestilib, which is a heat shock protein 19 inhibitor. So we looked to see what our DMG cell line cohort, their response was to these drugs in a dose-responsive manner. And prasinostat had relatively good inhibition with an IC50 range of approximately 0.3 micromolar, especially for our mutant cell lines. And we do have a little bit of an outlier there with our wild-type line, which is particularly resistant to prasinostat. When we looked at onalasertib, which is a heat shock protein 19 inhibitor, as I mentioned before, all of our DIPG cell lines were really sensitive with an IC50 range of between 0.04 and 0.197 micromolar. Onalasertib is actually really interesting because it has a heat shock. It has various chaperone proteins such as EGFR and PIK3CA, which were again two significant targets that came out of our CRISPR screen analysis. It also has an interaction with HDAC2, which again goes back to DIPG sensitivity to histone deacetylases, and has been shown in GBM models to penetrate into the penetrate through the blood-brain barrier and into the brain in both tumor-bearing mice and non-tumor-bearing mice. And a recent study has also implicated that it may also radiosensitize cell lines. So there is that combination with the current standard of care. So just to summarize, so from our research we've developed the largest single collection of DIPG-DMD patient-derived cell lines, and we've carried out very high-throughput multi-omic analysis and functional screening data. So from this pipeline, we've identified 22 molecular targets with both tumorigenic dependency and associated therapeutic sensitivity. And we validated HDAC2 as a H3.3 K27M specific molecular target in comparison to H3.1. We are currently investigating prasinostat, a pan-HDAC inhibitor, and onalasertib, a heat shock protein 19 inhibitor, in vitro and efficacy as single agents and both as a combination along with the current standard of care. But collectively, we have shown that the utility of this functional genomics pipeline is able to discern druggable molecular dependencies for therapeutic exploitation in DIPG. And this work would not have been possible without the support of a very wide team. So I do just want to thank the Developmental and Cancer Biology Group, everybody in the VPCC Next Generation team, our external collaborators and our funding bodies, the Children's Cancer Foundation and Hudson Institute for co-funding my PhD scholarships, and the Robert Connor Dawes Foundation and the Harold Mitchell Foundation for providing travel grants to be able to attend this conference. Thank you, and I'm happy to answer any questions.
Time for one question, Paul. I'm really interested in either side. Right? It is on. Either side of your Venn diagram, which is, did you get drugs when you deleted the target gene, you got no response, or where you deleted the target gene and it was dependent, but the targeting drug didn't respond? So your 22 targets are the ones in the middle. What were your surprises on either side?
Yes, so that is a question that we've always kept in the back of our heads. We don't want to dispute anything on either side of our Venn diagram. It's not something that for my project I've investigated too much in detail, other than looking at the different types of classes or what type of pathways those other drugs or genetic targets are involved in. I've tended to be a little bit narrow in looking specifically in the 22. I only have 3.5 years for a PhD. I probably need much longer.
Tons of time. We can make a PhD a bit longer.
No, I think I'm almost at my max at the moment. So.
Okay, we'll leave that there. Thank you. Thanks again.